Hello friends, today in this video I am going to discuss about the MCQs on immunology, and this is the 8th part of the series of more than 1800 MCQs on immunology with detailed explanations. Okay, and this video will be beneficial for many life science exams like CSIR NET or ICMR NET, GET Life Science or any other MSc entrance exams. Okay, so let's start the whole MCQ session with detailed explanation, and this is our Telegram channel where we can follow us. Okay, so now let's start the question answer session on immunology with detailed explanation. So the first question is, a monoclonal antibody has all the following properties except has antibody combining site that are identical or may be present in serum of patients with multiple myeloma, or can be obtained by hybridoma fusion technique, or produced by a single clone of T lymphocyte. So the right answer about a monoclonal antibody, where the all the properties, so all the properties except, the answer is option D. So a monoclonal antibody has all these properties except the last one, that is it cannot produce by a single clone of T lymphocyte. Okay. So the next question is, the earliest immunoglobulin synthesized by the fetus at 20 weeks is IgG, IgA, IgD or IgM. So the right answer is option D, that is IgM. So IgM is produced at the earliest, that is the earliest immunoglobulin synthesized by the fetus at 20 weeks. Okay. So the next one is, monoclonal antibodies are less specific than conventional antisera produced by a single clone of T lymphocytes secreted by myeloma, thymic lymphocyte hybridoma, or formed of a single class of immunoglobulins. And the right answer about the monoclonal antibodies is option D, that is it formed of a single class of immunoglobulin. Okay, and how we can form the monoclonal antibodies? So at first we have to inject a antigen into a mice. Okay, and the mice raise immunization, that means the mice uh, raise the antibody. Okay, so isolate the immune cells, that is the antibody containing cells, that is the antibody forming cells. So isolate them and then fused this antibody forming cell with the tumor cells. So after fusion it will be called the hybridomas. Okay, and this hybridomas screened for production of desired antibodies. So this hybridoma, this produce many antibody producing hybridoma clone. Okay, and in this, and from this we can isolate the monoclonal antibodies. Okay, so this is the procedure of producing monoclonal antibodies. So move on to the next question, that is, an example of autoimmune disease is brucellosis, tuberculosis, pernicious anemia or meningococcal meningitis, and the right answer is option C, that is pernicious anemia is a autoimmune disease, and this pernicious anemia is a decrease in red blood cell where the body can't absorb enough vitamin B12, and it is an organ specific autoimmune disease in which the body's immune system attack the lining of the stomach. Okay, and it is considered as a deadly disease due to the lack of available treatment, and pernicious anemia is most common in uh, Caucasian persons of North European ancestry than in other racial groups. Okay, so now the next question is, the lymphokine which protects cells against virus infection is chemotactic factor, transfer factor, gamma interferon or alpha interferon, and the right one is option D, that is alpha interferon. So the lymphokine which protects cells against viral infection is alpha interferon. And what is lymphokine? So lymphokines are a subset of cytokines that are produced by a type of immune cell known as lymphocyte. So they are protein mediators typically produced by T cells to direct the immune system response by signaling between its cells. Okay, so the next one is low molecular weight substance which do not induce antibody formation but cause delayed hypersensitivity is proantigen, cryptantigen or hapten or complete antigen, and the right one is option A, that is proantigen. So low molecular weight substance which do not induce antibody formation but cause delayed hypersensitivity is proantigen. So what is delayed hypersensitivity? So delayed hypersensitivity reaction, also called type 4 hypersensitivity reaction or cell mediated hypersensitivity reaction, which is a inflammatory reaction initiated by T cell, that is T lymphocytes, and antigen presenting cells such as macrophages and dendritic cells that cause an inflammatory reaction to either exogenous or autoantigens which take more than 12 hours to develop, and that's why it take long time, that's why it is called delayed hypersensitivity. Okay, so the next question is, the antigen combining site on immunoglobulin molecule is carboxy terminus, amino terminus, hinge region or H region, and the right one is option B, that is amino terminus. So the antigen combining site on immunoglobulin molecule is amino terminus. So this is the carboxyl terminal, this is the amino terminal. So the antigen binding site is present in the amino terminal. Okay, so here antigen binding site, this is the antigen binding site, this is the antigen binding site where antigen binds. Okay, and this is the amino terminal end of the immunoglobulin. So move on to the next question, that is, thymus-specific antigens are present on the surface of phagocytes, macrophages, B cells or T cells, and the right answer is option D, that is T cells. So thymus-specific antigens are present on the surface of T cells. Okay. So next question is, papain breaks down IgG molecule into one FAB and one FC fragment. So one FAB means fragment of antigen binding, AB means antigen binding, F means fragment. So one FAB and one FC fragments. FC means fragment of constant region. Okay, and or two FAB and one FC fragment, or one FAB whole two fragment or two FC fragments. So papain breaks down IgG molecule into option B, that is two FAB and one FC fragment. So here we can see this is the whole intact IgG molecule. So when we add papain, so papain break the immunoglobulin or IgG molecule in this region. Okay, so we will get two FAB region and one FC region. So the option B is the correct. Okay, so move on to the next question, that is, IgG antibodies have molecular weight of 9 uh, 90 90 lakh Dalton or does not cross the placental barrier or are present in secretions, take part in classical complement pathway. Okay, so the right answer about the IgG antibodies is option D, that is the IgG antibodies take part in classical complement pathway. So what is classical complement pathway? So here, so in this here the microbes, so if a microbes encountered in the body, so with the so the antibodies come and bind with it, and in the microbes and antibody complex, here the fast protein C1 will come, and after that consequently at the end in the classical pathway the cytosis or cell lysis will done, that is the microbes will be removed from the cell, from the body. Okay, so this is the classical pathway of protection, okay, or immune system. Okay, so move on to the next question, that is, IgM antibody is a monomer, dimer, trimer or pentamer, and the right one is option D, that is pentamer. So IgM antibody is a pentamer. So here we can see this is the pentamer of antibody, and here they are connected with each other by J chain. Okay, so move on to the next question, that is intravascular distribution of IgM is 45% or 50% or 75% or 80%, and the right one is option D, that is 80%. So intravascular distribution of IgM is 80%. So intravenous means inside the vessel. So this intravascular means situated or occurring within a vessel or vessels of an animal or plant, especially within a blood vessel or blood vascular system. Okay, so move on to the next question, that is half-life of IgG is 2.3 days, 5 days, 6 days or 23 days, and the right one is option D, that is 23 days. So half-life of IgG is 23 days. And the next question is Octernoni procedure is single diffusion in one dimension, double diffusion in one dimension, single diffusion in two dimension or double diffusion in two dimension, and the right answer is option D. So Octernoni procedure is a double diffusion in two dimension. So here the Octernoni procedure is, here a antibody is placed in a agarose gel, and here in two wells different types of antigens are added. So antigens will diffuse in this here and antibody will diffuse here. Okay, and they produce a antigen antibody complex in a certain region. Okay, so if we get a full identity or a continuous line like this or like this, then we can say line of precipitation at their junction forming an arc represents serologic identity or the presence of the common epitope in antigens. So these two antigens are common, they have common epitopes. Okay, so if we get non-identical lines like this non-identical lines, then a pattern of crossed lines demonstrate two separate reactions and indicates that the compared antigens are unrelated and share no common epitope. So these two antigens are not same or their epitopes are not same. And the last one is if we get a partial identity, so like this is the partial identity. So if we get a partial identity, then we can conclude that the two antigens share a common epitope, but same antibody molecules are not captured by the antigen and transverse through the initial precipitin line to combine with additional epitope found in the more complex antigens. So they are the same antigens but they have different epitopes. Okay, so this we can conclude from the double diffusion technique, that is the Octernoni double diffusion antigen antibody complex technique. Okay, so move on to the next one, that is tumor necrosis factor alpha. So the tumor necrosis factor alpha is it promotes division and differentiation of monocytes or stimulates expression of class 2 MHC antigens or augments NK or natural killer cell activity or promotes granulocyte differentiation, and the right one is option C, that is augments natural killer cell activity. So tumor necrosis factor alpha augments natural killer cell activity. So the tumor necrosis factor alpha, that is TNF alpha, was initially recognized as a factor that causes the necrosis of tumors, is naturally produced by activated macrophages and monocytes. The inappropriate or excessive activation of TNF alpha signaling is associated with chronic inflammation and can eventually lead to the development of pathological complications such as autoimmune disease. Okay, so move on to the next question, that is all are phagocytic cells except macrophages, Kupffer cells or astrocyte or microglia, and the right one is option C, that is astrocyte. So all are phagocytic cells except astrocyte. And these astrocytes are star-shaped cells found in the brain. Similar to other neuronal cells, astrocytes are comprised of synapses or cell ends that allow for chemical and electrical communication between cells. Okay, so these are the astrocytes. These are the astrocyte cells which are found in brain. Okay, or neurons more specifically. So move on to the next question, that is lymphokines are produced by dendritic cells, stromal cells, or B lymphocyte or T lymphocyte, and the right one is option D, that is T lymphocytes. So lymphokines are produced by T lymphocytes, and lymphokines are a subset of cytokines that are produced by a type of immune cell known as lymphocyte. They are protein mediators typically produced by T cells to direct the immune system response by signaling between its cells. Okay, so move on to the next question, that is primary immune response has a negative phase, has a lag period, or antibodies are mainly of IgG class, or has a rapid rise in antibody titer. And the right one is option B, that is has a lag period. So primary immune response has a lag period. So next one is IgM antibodies. So IgM antibodies have 160,000 molecular weight, have a sedimentation coefficient of 19S, or can be transported across the placenta, or are long lived and disappear later than IgG. And the right answer about the IgM antibodies is option B, that is have a sedimentation coefficient of 19S. Okay, so next one is T lymphocyte. So T lymphocyte bind to ship erythrocyte antibody and complement forming EAC rosette, or has C3 receptor on their surface, or do not have thymus specific antigens on their surface, or undergo blast transformation on treatment with canavalin A, and the right answer is option D. So T lymphocytes undergo blast transformation on treatment with concanavalin A. Okay, so move on to the next question, that is complement is a complex of 9 different fractions, is not normally present in the serum, or constitute about 80% of the human serum globulins, or does not get inactivated at 56 degrees Celsius within 30 minutes. And the right one is option A. So complement is a complex of 9 different fractions. So here we can see the whole complement pathway are three types of complement pathways are there, that is classical pathway, lectin pathway, and alternative pathway. So all total here we can see C1, 4, C2, C3, C4, C5, C3, C5, C6, C7, C8, C9. So all total 9 fraction of different complex proteins are present in complement. Okay, so the option 1 or option A is the correct answer. So move on to the next question, that is antigen presenting cells are all phagocytic cells, are capable of presenting antigens to plasma cells, or include B cells, dendritic cells, or Langerhans cells, or macrophages, or are B cells only. So antigen presenting cells include B cells, dendritic cells, Langerhans cells, and macrophages. Okay, so move on to the next question, that is human leukocyte antigen complex induces complement activation, controls T cell recognition, and elicits allograft reaction, or is involved in the adherence to endothelial cell surface, or is involved in platelet aggregation. And the right answer about the human leukocyte antigen complex is option B, that is it controls T cell recognition and elicits allograft reaction. Okay, now the next question is Retter syndrome is a triad of conjunctivitis, arthritis, dryness of mouth, or conjunctivitis, urethritis, or arthritis, or conjunctivitis, urethritis, or dryness of mouth, or urine urethritis or arthritis or dryness of mouth, and the right answer is option B, that is conjunctivitis, urethritis or arthritis. And this triad means a group or set of three related people or three related things. So here three related disease. Okay, so Reiter's syndrome is a triad of conjunctivitis, urethritis or arthritis. Okay, so the last question is, cell mediated immunity is not responsible for arthrus reaction, contact dermatitis or graft rejection or tumor rejection. And the right answer about the cell mediated immunity, so cell mediated immunity is not responsible for option A, arthrus reaction. That is, an arthrus reaction is refers to an acute localized inflammatory response that typically occurs after vaccination. So it is classified as a type 3 hypersensitivity reaction, which is when antigen antibody clusters, also known as immune complexes, are formed due to an abnormal immune system response, and the reaction is usually present at the site of injection after a vaccination. Example, tetanus diphtheria booster. Okay, so this is the all question answer of this video. So thank you for watching this video. So we will see you on the next video at our part 9 of immunological MCQs. Okay, thank you for watching this video.